Good morning, my dear students. I hope you all are fit and healthy. So, dear students, today we will study about Ascomycetis, which is also known as sac fungi. So, Ascomycetis is a large group with more than 30,000 species known. And this Ascomycetis, it includes diverse forms such as yeast, pink mold, brown mold, and other edible morels and truffles. So, my dear students, let us learn about some of the general characters of Ascomycetes. So, first thing you should know that they are mainly terrestrial and they occur throughout the year as saprophyte, means they will be feeding on dead and decaying matter, or they may found as parasite, means they will be living inside or on the body of the living host. So their body is haploid, means they have haploid nucleus, and their mycelium it is branched or septate. My dear students, their most important feature is the formation of a sac-like structure that is known as ascus, in which there are 8 haploid ascospores are formed. So this ascus formation, it is the most characteristic feature of this group. So here in this picture you are observing the two parts of reproduction. The upper part, it is shown the asexual method, that how the fungi, that is Ascomycetes, it is reproducing by forming the asexual spore exogenously on the outside of the surface of hypha. The asexual spore, that is conidia, is formed. So conidia is formed exogenously on the hypha. This is the asexual method of reproduction in Ascomycetes. But when the fungus, that is Ascomycetes members, they have to reproduce sexually, then what happened? They are forming a sac-like structure that is known as ascus, in which there are 8 haploid ascospores. Now the question arises how this ascus is formed. So one of the conidia, when it fuses with the another hypha, so now there will be formation of a sac-like structure that is known as ascus. So in the ascus there are two haploid nucleus present, one from the conidia and other from the other partner. So here in the ascus there are two haploid nucleus present. Now these nucleus, they are in a dikaryon phase, means they have not fused yet. They will be kept as such in the ascus. In most of the members of this Ascomycetes, the dikaryon phase, it is continued for many years. Now when the two haploid nucleus, they will fuse, they will form a diploid nucleus in the ascus itself. Now this diploid nucleus, it will undergo meiosis process. As you know that after meiosis there are 4 haploid nucleus will be formed inside the ascus. So in the ascus now there are 4 haploid nucleus again, and this ascus, they will again undergo mitosis, and this will result in the formation of 8 haploid ascospores. So 8 haploid ascospores, they are produced now in the young ascus. So my dear students, you should always remember that no single ascus is produced. Ascus, they are always formed in groups. So, and these ascus in group, they are kept in a fruiting body that is known as ascocarp. So under favorable conditions, these ascospores, which are haploid nucleus in the ascus, they will be released and they will germinate, and ultimately they will produce the haploid body. So basically what happens is that each of the nucleus in the ascus, they will be surrounded by a thick cell wall now, and cytoplasm is also there. So these are now known as ascospores, and these ascospores, they will be released from the pores or through slit, and they will be dispersed by air current, and under favorable condition they will form new mycelium. So entire what is the use of this Ascomycetis? So the Ascomycetis has a very large economic importance in food industry, in medicine industry. So entire brewing, bread and cheese making industry depend on yeast for fermented products. So yeast, which is a member of Ascomycetes. So my dear student, this was all about Ascomycetes. So let us learn about the another group under this fungus kingdom. Another phylum is Basidiomycetes. So Basidiomycetes is also a large group of fungi with over 25,000 species, and they are the evolved fungi, and this group gets its name from Basidium, that is a club shaped structure at the tip of their reproductive hyphae, which will bear spores at its tips. So let us see how the spores they are produced and how the Basidium is formed. So before studying that, let us know some of the another important characters. So their mycelium, primary mycelium, it is short, but secondary mycelium, it is very large, vast. Primary mycelium, it is short-lived. However, if we consider the secondary mycelium, it is large, it is profusely branched, and there are septate, and it is also binucleate structure. So if you observed in the picture, the visible part of the fungus, it is known as Basidium. This Basidium, it originates from a binucleated structure where karyogamy and meiosis as occur. So this Basidium, it bears basidiospores on the outside, and their life cycle, it is characterized by short diploid stage. So the basidiospores, they contain single haploid nucleus, and these spores, they germinate to produce a monokaryotic mycelium. During the fusion of this monokaryotic hyphae, the nuclei of one hyphae move to the other, and this hyphae is known as dikaryotic, and each of these dikaryon is having two haploid nucleus. So the most important member of this Basidiomycetes is mushroom, the edible fungi. But some of the members of this group, they are cause worst plant diseases like rust and smut. Some of the examples are Agaricus, Amanita, and Patsenia, which causes rust disease in wheat. And the last phylum, my dear student, is Deuteromycetes. Here, most of the members of Deuteromycetes, they are in fact the Ascomycetes, in which the sexual reproduction is absent. So this is all for today. Have a nice day, everyone.